I'm just going to organize my screens. I hope everything here is working properly and people can see this. And
yes, we can see it great.
We will just go straight in. So this morning, I want to talk about one of my pet subjects of the last few years, about using natural intelligence in drug discovery, and how I think this is very important in looking for new antimicrobials and addressing AMR, which of course is a huge problem worldwide. So, just a rough running order is a little bit of philosophy. Then we'll come to a butterfly effect, when you'll see what I mean by that. And then we'll look a little bit at the history of antimicrobial discovery and thinking about a discovery void that you will see in the slides. And then I would just look at things reflecting on practices and the transition from target data, which became very much the vogue in the 1990s, and then looking at how you actually kill bugs. Then we'll consider a little bit about models, data, and changing practices, and consider physical properties. And then we'll talk a lot about permeability and permeation and why this brings compromise in drug discovery and how nature might help us there. And this brings us to, considering natural intelligence. So that's kind of a summary, and we will meander through these themes as we go through. So I like to think about rules and guidelines in drug discovery, and Mike Han always told me that rules are for the obedience of fools and guidance of the wise. And I also like my favourite guerrilla artist Banksy, who talks about the greatest crimes are not committed by people breaking the rules, but people following or enforcing them. And, a lot of drug discovery is probably guilty of that. And a couple of years ago, I read an old friend of mine, Robert Hannigan, who's now warden of my old college in Oxford. He was head of GCHQ in the UK, which is our kind of spying agency, monitoring communications and code breaking. And it's kind of the legacy of Alan Turing and the Bletchley Park code breakers. And that's kind of the background of the story in this. What can the secret world teach the rest of the world and big business about problem solving and creativity? People might say that code breaking is rather easier than the impossible task of drug discovery. And actually, a computer was developed to help this 80 years ago. We will hear about computers in drug discovery a lot as we go through. But one of the things he really talks about here is the importance of the diversity of thought. And it comes through that groupthink, when we're all doing the same thing, will hinder progress. So you need lots of ways of considering problems in code breaking. So my butterfly kicked off a couple of weeks before I left GSK. I took this lovely picture of a white letter hair streak butterfly. I was spending time in a place for nature, and it reminded me that very soon I'd have a lot more time in such places. Natural history is one of my many great passions. So if you want to see more photographs, that link will take you to my website. And to go quickly on, I kind of put a consortium together with some friends, old friends, new friends, and people I'd done some work with. And we looked at, this paper was kind of a background and just a bit of a thesis for what you might understand in drug discovery. And one of the premises we said that if a compound is made by a protein, i.e. an enzyme, and Nick and Sabina were experts in enzymatic synthesis, you're likely to be transported by a protein and you could inhibit another protein, your target. And we will come back to this notion of transporters and getting things through biological membranes. And, natural motifs in drug discovery are very important, and we will come back to this. And this brought through the notion of natural intelligence, and truly organic molecules are not petrochemicals. And we will revisit this. I should say that many of these slides I will go through relatively quickly, but the links are there, the slides will be shared with people. So if you want to go back and read things in more detail, please do so after the talk. Sorry, my mouse is playing me up here now. But that is working. So one of the key things, let me just check I've not gone 2 slides, that we showed was how much nature has been disappearing in drug discovery. So these are data that Paul Leeson applied to his data set. He used the natural product scout algorithm, which is referred to here. There's also another one that Peter Ertl produced, and it gives a very similar outcome. We can see that the natural class, so that the probability of compounds being or looking like a natural product, has really tailed off, especially since about 1990 and the days when high throughput screening came in. We know that there's this drift up in the molecular weight of drugs. We will talk about that in due course as well. But we've seen that, by and large, lipophilicity may have edged up a little bit, But this is log P, log D, maybe doesn't show such a big trend, but we can see there's a relatively narrow window on this. What has changed a lot in drug discovery has been the switch over to heterocyclic compounds and aromatic nitrogen atoms. People have seemed to have to employ these probably to control lipophilicity, and that's discussed in later slides. So nature is going down in drug discovery. And when I published this, my friend Julio pointed out that we were thinking with natural intelligence, and he introduced me to this wonderful book by Lean Gorison, which, a bit like Robert Hannigan's book, looks at things from a different perspective, and it looks at many aspects of life, organization, how we treat the environment, could be addressed differently if we considered how nature does things and what nature has evolved over billions of years to do. I found this as a nice picture of evolution. And if you look back to the age of the Earth and when the sun starts up, 4 or 5 billion years, we then come to this point where life starts to appear on Earth, with the very simple organisms getting a little bit more complicated. And of course, we're thinking about bacteria, protozoa, and some of the other kind of organisms that we're seeking to treat in neglected diseases and in addressing AMR. So these fellows evolved over 2 or 3 billion years, and they still have had a long time to evolve in in the billion or more years that have gone since, and natural selection and natural processes. If you can think about it, life is something of a biochemical arms race. Some compounds, nourishment, sugars, proteins, etc., and fats, we need these to exist, to thrive. We also need some of these things to defend ourselves. Ultimately, a lot of the defensive mechanisms will come down to chemistry. And if you can evolve those, change color, you can put poisons inside you, they will stop your predators from having you. But of course, all of this, there's kind of, when us humans came along and when we learned to write, is just in this blink of an eye at the end of this chart. And even more so, the science of killing bugs with bacteria. This was me after delivering a lecture at my alma mater to the iNOS Institute, where actually I've been doing a training course, and it was a great irony and a great pleasure to give these talks in the Dunn School at Oxford. And these are the very buildings where Florey, Chain, Heatley, and others first isolated enough penicillin to be an effective medicine in the beginning of around the beginnings of World War II. And you think of this, that's just 80 years as opposed to 8 billion. And again, I won't go into detail on this. I'm kind of using this for illustration. This shows you what has happened in those 80 or so years since penicillin and all the new antibiotics came along, and this sort of so-called golden age up until the 50s, 60s, when many new agents were discovered. We can see the genesis of compounds, and of course along the bottom we can see the issues with resistance coming in and ways that we've had to counteract this with using combinations and different agents. But what you'll notice, for those of you who know your antibiotics, many of these, or the majority, are natural products and their derivatives. Another insight onto this will come from this nice picture I found on this React database or website. And they talk about this discovery void, which we saw on the previous slide as well. It shows you when new classes of antibiotics, different structural classes, different modes of action, were discovered. And it talks, this paper is a little bit old now, but it talks about that discovery void, probably 1990, 2015, where no new mechanisms were discovered. And I just wonder, is this coincidence in terms of the rise of high throughput screening and different mechanisms that were being used in drug discovery? It's not all doom and gloom. My former colleagues managed to bring the first topoisomerase inhibitor, or a new topoisomerase inhibitor, to the market. So things have changed, and there were some change in practices to deliver this. So now that I'm no longer a corporate person, I can actually question, what people were doing. And it used to break my heart when I was having to go through the depths of a high throughput screening program. I think that high throughput screening causes many problems in the industry, and as many metrics, as many new methods and methods of sorting out what you've got from your screen as it's solved. Drug discovery leaders loved it because they were fixated by numbers. How many compounds did people make? Could we make 2 million compounds and get one drug out? These kind of numbers. People were addicted to potency, and I'm afraid some people in the industry still are. There's that idea that you could pour 2 million compounds into a high throughput screen, you could get a hit, and it would take a long time sometimes to qualify that. Whole areas became sciences because of the practices. Had to do hit to lead chemistry, lead optimization. This was the typical industrialized paradigm for discovering drugs. And this paper here will talk to you about the issues of primary research translating into the clinic. And we all know about how few compounds come out of this funnel to do it. My friend Mike coined the term molecular obesity, potency, and other addictions in drug discovery. And that kind of nicely dovetails with things that will follow. But you don't have to do this in an industrialized way. There's 4 Nobel Prize winners and a guy who did a fantastic job in academia here, and big selling medicines that were discovered by what I would like to call in cerebro drug discovery. So, they haven't used in vitro methods, in silico methods, all of these, reductionist ways that people tend to use in drug discovery now. 2UU says every scientist dreams of doing something that can help the world, and her life's work led to artemisinin to treat malaria. Rick has often done structure-based modifications to small amino acids. I kind of interacted with Rick back in my iNOS days 25 years ago. But Lyrica, the kind of with the active ingredient pregabalin, came from his systematic study of modifying amino acids, and this is still one of the biggest selling medicines to this day. Recently, Caitlin Carrico, base modified DNA, RNA. This was the very key part in the immunogenicity of the RNA vaccines, but just fundamental research and just fundamental small changes enabled all of the progress there. But I should sort of pay tribute to, I had the great pleasure of working a few times with Trudy, a truly wonderful person. George Hitchens was a little older, but these guys were up there with the greatest drug hunters of all time. I think Dennis Liotta says he's getting close, but he was very much a nucleoside chemist. I'd look for many of the things he did. But they mimicked the metabolism and the anabolism of purines and pyrimidines to make various medicines. And they were, these are ones that they're part of. You could argue that AZT is kind of part of this area, and other things came in their legacy. But they're always looking at natural products and their modification in the way they did things. So in cerebro drug discovery. So how does this fit with antimicrobial work? This is a paper 10 years old now from AstraZeneca, and they were looking at the lessons that could be learned from the labyrinth they'd found themselves in antibacterial drug discovery, and how difficult they'd found it to find decent medicines that gave broad spectrum antibacterial activity. And most of the time, this was potent compounds against the target, but they couldn't get this antibacterial activity. And they ascribed many of these difficulties to the problems of understanding the efflux systems. So here's your cartoon of your bacterial cell walls that I'm sure many of you are familiar with. The similar things will apply to other parasites. It's just understanding how you can get molecules through this and then keep them in there without falling victim to efflux mechanisms to do the job you want your compound to do. So it gives you some of the issues in this area. I looked at a couple of graphs in particular in here and some of the issues that the way that the research was done might bring ahead. This graph here kind of shows you the average lipophilicity in terms of the log D of the compounds that were found in the screen. So these are kind of the starting places that people would find from their screen. The numbers are down here. These are different isolated gene antibacterial targets. This graph looks at the particular organisms, and it looks at the difference in terms of the compounds with a decent MIC, the actives, versus those that didn't have a good, that didn't give you an antibacterial activity. Many of these would have activity in a bug. And you can just see this disconnect between the lipophilicity, i.e. quite hydrophilic compounds that would tend to give you an MIC versus the many compounds people were looking at. This graph as well, it shows you one particular program, and it shows you with time how there is this drift up in lipophilicity. The more red, the more orange the compound, the higher the lipophilicity is to get decent activity. And there are most of the green compounds have a very limited IC50 against this particular target. So it's this idea of translating the fundamental activity against the target into activity against the bug. We will come back to that. Former colleagues of mine at GSK, they also looked at things and some of the issues. This is a rather older paper, in a retrospective of 70 high throughput screening campaigns that basically didn't show very much in terms of decent activity. And it talks about the change in looking at compounds that might have what you might call rather more drug-like properties, which is a term that I've come to dislike. Their properties akin to those of many drugs is a way I would like to use that term these days. But they talk about the importance of looking at the critical path pharmacology, the MICs, and the microbiological efficacy as important things in looking for drugs. And it still showed that even with this, there was still quite high attrition. We will come back to this graph, but this is from that paper where it starts looking at what you might call a Lipinski type analysis in terms of lipophilicity on the y-axis and the molecular weight of compounds. They claim that CNS compounds are representative of many drugs, and you can see that the blue triangles by and large are all up here. And you start looking at the size and especially lipophilicity of many antibacterial classes. You can see that there is this disconnect, and they're quite distinct from the other compounds. So we'll take a little drift away from this now, and I often like to use this quote that again, this is a Mike Hannism that he introduced me to George Box, who published this paper 40 years ago or 50 years ago, saying that all models are wrong, but some are useful. So anything you fundamentally do to describe a process is never going to be perfect, but many are useful. When you have these models, though, he says you need to be somewhat parsimonious, that if you put too much elaboration, too many parameters, it can over elaborate and it will confuse you in the model. And he tells you that you should follow William of Wellcome, so Occam's razor principle, that you need to get things that are very simple and just look at simple descriptors that can help you understand and get some insight on the phenomenon that you're modeling and the relationships you're looking for. And he also warned that if you worry selectively, that since all models are wrong, you need to be alert within that model what is importantly wrong. And he sort of uses the quote, it's inappropriate to be concerned about mice when there are tigers abroad, or maybe it's mice and the lions are more prevalent in South Africa. And I kind of looked at this in terms of the rule of five, and I suspect Franco is on charge, the second author on the infamous paper. And I was asked to write a review, having published a few things over the years about the rule of five. And you have to look at it in the context of when it was made. It was made in the 1990s when everyone was addicted to potency and high throughput screening, more lipophilic, bigger, tended to give you more potency. And Chris and Franco and people noticed that when you were doing this, you weren't getting the translation to get good permeability and solubility. So when you read the paper, it's all about permeability and solubility. It kind of got conflated with drug-likeness at a later time. So in a nutshell, I don't think that weight matters in drug discovery. And most things you see now that are purportedly beyond the rule of five are just heavy. The molecular weight is higher in general. You will see that lipophilicity is the most important thing, but lipophilicity has moved on since C log P in 1997. We can get high throughput measurements, and measurements are what matters. And I believe it's log D, not log P, that matters. As you get bigger, you need to put your number of acceptors up just to keep your lipophilicity in check. We saw about aromatic nitrogens earlier. We may come back to them. Donors do matter, but they don't always matter. It's a good rule to minimize donors, but sometimes you can get internal hydrogen bonds, you can get a carbohydrate with many protonated oxygens. They can happen, they can help you. But that's a nutshell, just to describe. But one of the quotes I like to think is that in times of profound change, the learners inherit the earth, while the learned find themselves beautifully equipped to deal with a world that no longer exists. So there was a huge amount of change with high throughput screening, and I kind of lived through this when we moved from Glaxo to Wellcome, and we all had to do high throughput screens. You know, if we didn't make 100 compounds a week, we were nobodies. But I kind of always was a little bit against that. I wanted to get compounds that mattered. And what it's telling you that if you don't look at things and learn them, you do your old practices, you're kind of falling behind, and you need to equip yourself how to work with this new world. But you know, this is nearly 30 years ago now, and a lot has changed in drug discovery in terms of targets and understanding since then. So these guys were the learners in the late 1990s, and you know, by and large, it does stand the test of time, except where compounds have a high molecular weight. But what's really important in there is there are, you know, discrete statements about the role of transporters in drug discovery. So, you know, think of it in this way, that molecular weight, I think, is the mouse in terms of George Box's description. Lipophilicity is the tiger. And if you look at chromlog-D, which is a modern measurement of lipophilicity, it's a very good tiger detector. And there are references and slides on chromlog-D in due course. But what you must remember with the great work that these guys did is that it's all based on probability and 90th centile thresholds. So the fives aren't thresholds. It doesn't mean to say that you've got a molecular weight of 498 and a log P of 4.8 that you're compliant. That means that you're in the, you know, doubly in the minority, if you like. And this is a paper I wrote with Paul Leeson explaining this. So do think about these rules and how you interpret and use them. So we go back to this slide we saw before, and I kind of look at this in terms of, you know, these are the boundaries, and these and you know, this is the distribution in terms of lipophilicity and molecular weight where antibiotics are found. We've got this discovery void that is overlapping with high throughput screening. And this is an Einstein quote I like to use in this context. You know, we cannot solve problems with the same thinking that we use to create them. And you know, my thesis here is that did high throughput screening, what we were putting into these screens, create the problems we have with antibiotics? So how can we consider this in a different way? Yeah, we have to consider what a good drug looks like. It's the multi-billion-dollar question, and what can we do to enhance or save lives? And I just love this quote from Corwin Hanks, the father of many things in lipophilicity and QSAR, and Al Leo and their team. And they said nearly 40 odd years ago, that without convincing evidence to the contrary, drugs should be made as hydrophilic as possible without loss of efficacy. And the key word there being efficacy, not just potency. And this is the essence of LLE, the difference between potency and log P. There isn't much in there, but there are other papers, and you know, there are references in here that go into this in more detail. But in reality, there are no simpler or prescriptive answers beyond this principle. Different drugs have different roles, different methods of administration, and you know, we will see some principles to follow. The big thing about drug discovery, it's a process of optimization, and you often have to make compromises. And the one thing that underlies all of this is the use of physicochemical properties. This is how you can measure and understand where you're going. So we all know that we have to do all of these things. You have to keep all of these plates spinning at one time. And you know, these are arbitrary descriptors of chemical space, if you like. That, you know, we've usually got some kind of lead that has potency, and we have to turn it into a drug where all of these things, safety, absorption, stability, solubility, you know, and other things, we have to get right in our molecule, and we have to make compromises to get there. This is a nice chapter in a book that I contributed to. It's a very nice guide that Patrick Schneider was the editor of, where he looked at many things that impact the ADMET, so you know, the ADME, so this is pharmacokinetics and toxicity of compounds. And this is just a snapshot from it of all the things that were looked at. They're all issues in drug discovery. And if you go through it, virtually everything that you do in drug discovery, it's made better if you reduce lipophilicity, with exceptions, as you'll see shortly. So, you know, ensure you can control that lipophilicity, as Hans said. This is a rather busy side. We've seen this before, that if you become more lipophilic, your potency is often reduced. It's a difficult thing. But your safety is enhanced, your solubility is enhanced, and your metabolism goes down if you reduce lipophilicity. You may be reduced in terms of your absorption. And this was a graph I produced 20 odd years ago now, or about 20 years ago now, looking at the impact of PFI. So this is a thing I developed with Alan Hill at GSK, where we added the aromatic ring count to lipophilicity. These are probabilistic scores. These are all based on real data, and it shows you risk levels with different, if you like, developability assays in terms of solubility, protein binding, P450s, clearance, permeability. We also looked at hERG and promiscuity, and you can see that by and large, the greens are over here. But we've always got to make this compromise in terms of permeability. And we can see that as we get to a place where we get better permeability with compounds, we start getting into ADME constraints. We start getting bigger risks with these other things. And we've got more risk of off-target effects as we get to the best place for permeability. Has nature evolved a means that when you're starting to get more permeable, your P450s will kick in to turn the compound over, for example. And I hope that most people have read the publications that if you get very lipophilic and very aromatic, then your chance of success is so much reduced. And you need to have a portfolio of compounds where you're not working in this area to give you a better chance of not failing. So permeability is where we're coming now. And this is a subject of a lot of debate in the literature, especially about 15 years ago when Doug Cale was putting forward his theories that everything goes through a membrane on a carrier protein. Nothing will partition between outside the membrane into the bilayer of the membrane through this charged polar tail, and then you have to get back out the other side to get into the cell. He would suggest that everything will go through via a protein or a transmembrane body that will help you get through your membrane. It's a little bit like partition, and we will come to that in a second. We partition in, but this is only a two-way thing. This one has a double partitioning. This group, led by some academics and some people in industry, they acknowledge that carrier-mediated processes are there, so that is no doubt. But they do suggest that passive is also important. But whether that passive is through the membrane or through some generic proteins is a matter of debate. The one thing I don't like about this team is the only thing I've ever seen them rationalize their observations is by the rule of 5. And as you will see, I don't think it's appropriate in some things with permeability. So these are real data. This is actually from one of that academic industrialist team's paper, not Doug Cale's, where they look at the kind of the mass transfer across artificial membranes against measured lipophilicity for a number of drugs. And you can actually see there is this bilinear relationship. These are data from our own papers where we show that the optimal passage through a pamper membrane, and it's very similar in a cell-based assay, has a maximum at a log D of about 3, or this is 5 on the chromatographic scale. The grey bars are where the data failed because there was poor recovery of the insoluble compounds, or they stick to the plastic. So I think it's right in there to show that these compounds were part of this analysis. And that just does really show you this nice bilinear response between lipophilicity and permeability. It's just very simple. If your compound is very hydrophilic, so you've got low log D, you will just stay in the aqueous layer this side of the membrane. As lipophilicity goes up, you will tend to go in there. If you're too lipophilic, you stay there. You don't come out very quickly the other side. And this will be the same, be it going through the membrane or through a more generic protein target. So that's kind of the basics for doing this. Other people have done similar things. This is some of Scott Lokey's work, looking at some of the peptides and the analysis of the impact of lipophilicity, what happens there. And we can see this log P of 3, equivalent to 5 on the chromatographic scale. And we can see where the best permeation comes from these. You can also see that when compounds are more lipophilic, there's very poor recovery of the compound between the different wells. So this is looked at. I don't have reference to the AbbVie MPS here. but this kind of behavior is a key part of understanding permeability of the big compounds that they looked at. So how do compounds enter in cells? And these are some pictures I stole from Doug Kell, and he just says that if you actually look at the average membrane, there isn't very much bilayer exposed. It is just full of all of these transporters. There's a ratio of protein to lipid of about 3 to 1, and this will show you, where some things are known to come in and where some things are known to be fluxed out. So this is the real situation and how molecules might go through. There are talks, there's a supplementary slide about the importance of this, looking at exogenous natural products. And it seems that many of these carriers evolved to bring beneficial things into cells from our diet, but I haven't got time to go that in detail. So let's consider this in terms back of our microbial membranes and these transporters and the ways of going through these, really thick, waxy cuticles. So if we want to get through there, we must mimic something that the bug requires to be, enabled to get inside it. So is it something nutritional or a cofactor that it needs to suck in to survive? We also have to remember with bugs that this diet might change within a life cycle. TB hides out in different places and uses different sources of carbon to survive. Not only do we need to do this, but we need to escape the efflux mechanisms and other potential metabolism that can defend against this process. And the thesis here is that 4 billion years of evolution have helped design compounds that can escape this labyrinth. And this is where natural products come from. So this is a variation on the rule of 5 analysis from some work I did with Chris Timworth 5 or 6 years ago. If you look at a set of compounds, and these were Mike Schultz's compounds that I annotated, and he was looking at the rule of 5 and looking at that most things are actually compliant. But I actually went through this in a little bit more detail, and you'll see I look at it in a different way in subsequent slides. But I started colouring and classifying some of the compounds. When you look at amino acids, beta-lactams, carbohydrates or glycosylated compounds, nucleosides, peptides, different classes of drugs. And you can actually see that many of the so-called rule breakers are taking us into new territory. And you can see this is the same one we saw earlier from the GSK group in that review. You can see that we're all down here in terms of the much bigger antibiotic set that they considered in their analysis. And this segues into what I believe is the most powerful model in drug discovery. It's evolved in GSK since about 1990, when Alan Hill, my good friend, kind of developed this as a 4-box model. And they then found that this line here, and these are kind of, if you like, the error bars either side of it, these describe compounds that have a good chance or a poor chance of being orally bioavailable. And I was involved 3 times at GSK in shutting down Pampa and some of the higher throughput permeability assays with CACO and MDCK cells, because this model was more predictive of permeability and actually also of oral bioavailability. It's coloured here in terms of Lipinski views that the green compounds have less than 10 acceptors. The red crosses are those that have more than 5 donors. And we're just starting to look that most of the failures, in inverted commas, are what you might call below the line. And you're hearing me use that expression. And this is kind of the genesis of that model. This is a different set of compounds. Now, this is the training set on which the model was built. And this looks at absorbance class. So the green circles are compounds with greater than 30% oral bioavailability. And I believe this one was a human set. The red compounds have relatively poor bioavailability. Statistically, this molecule is very powerful. In spite of the fact we can see there are many green compounds down here, and these are the ones that we believe are natural products and probably are transported. So look at this in more detail when you have time. But I do believe that this is a very important way of understanding permeability. And then this is that graph we saw before with the particular classes, and we're starting to see how the natural compounds are, if you like, all below the line. Another analysis I did was with the set of antibiotics, and I annotated the TB compounds in this. And you can start to see about where all the compounds are below the line. So this is the line you've seen before that discriminates good from bad. These are lines that will help you distinguish paracellular absorption, but that's a detail in the paper. But what is really important in here is the coloration is good and poor predictions, but the size of the diamonds and the circles relates to the natural product likeness of these compounds. And this is just using the NP Scout algorithm. So if you look, the size of most of the points that are so-called below the line are recognizably natural products. This probably needs to be repeated on compounds with with a bigger data set. I need to get myself a bigger data set and then get the calculations done on that. But what you also notice that some of the ones that have a lesser natural product likeness are also below the line. And we will come into pseudo-natural products in due course. And I worked with my friend Liz Fulham, and we looked at consideration of where you might need to go to find new TB drugs. Do you want to be in this region where you're transported? The purple spots on here are the current TB pipeline. And we know where antibiotics, I think they're the green crosses on here, and there are different ways of classifying the passage of compounds through membranes down to some of this recognition processes. And I think there is something in this, although there's still a little bit more work to do to prove it. And these are other kind of thoughts on it from the paper with Chris. These two are kind of rotated through 90 degrees, but you can see where good permeability is. Mike Cann and George Kedrick talked about a sweet spot. Pfizer talk about a golden triangle to go for. Small hydrophilic molecules are paracellularly absorbed, but that's a whole other lecture. And it's just understanding where you need to be to get this permeability, which is often the thing that's difficult in drug discovery. This is from Time and Place for Nature, and I'm just thinking as well that this is where antibiotic space is. It's quite different from this sort of classical sweet spot for drug discovery, where most drugs will be found. So just a kind of a final aside is just thinking a little bit with natural intelligence. So one of my colleagues at Wellcome used to have a poster of this on his board, and I found this just online somewhere, that God or the creator, whoever it may be, is at evolution. Only a few perfect heads were made. The rest were covered in hair. And I'm getting to the point where this is starting to apply to me. And I start thinking with natural intelligence, how many perfect molecules evolved? Were there perfect proteins, smaller peptides, and other molecules? And you often look, and Ben Davis at Oxford always bringing this to people's attention, that if you look at a protein structure, that is only part of the active compound you look at. And often they're absolutely decorated with carbohydrates and other post-translational modifications. So what are the impacts of all of these? So when you want to do AlphaFold and some of these predictive things with protein structure, what are the implications of the glycosylation state, the myristylization state of the compound? We all know how much money the drug discovery processes or the industry has invested in a single phosphorylation event, kinases. If one phosphate makes so much difference to the actions of a protein, what do these other molecules do in terms of the action of the protein? And could you be arguing that the same thing applies to small molecules? Are they natural? Are they recognized? Do they have post-translational modifications that help get you through membranes? And you start looking at drug discovery. This is looking at metabolic engineering to try and find new antibiotics. We know about the aminoglycoside type compounds. What is it about Taxol that's evolved to get it into the place where it does what it does? There's lots of, cyclosporin is always the poster child about chameleonic compounds. It can have different conformations depending on its environment. So what do we need to understand how these compounds act? And even just in the last couple of weeks, I've noticed a couple of things. There's this new antifungal peptide, which has extensive, it's got a lot of lipid added to one end of it. And this lipid has a big impact on the pharmacokinetics and the actions of this compound. It can, you see some wonderful structure-activity relationships and also structure-property relationships. Think of the Ozempic and all of these new wonder peptide drugs. They've all got these massive amounts of fat added to them to modulate their pharmacokinetics. So is there a role for these in antimicrobials? Another one I saw just this week, people were prenulating small drug molecules, and they were getting improved permeability with such structures. So these are almost like, if you like, post-translational modifications. I highlighted this one. I noticed this a few years ago. A new antibiotic was described in Nature, where when it was discovered, people looked as much at the structure permeation relationship to go through a pouring. They looked at that every bit as much as they looked at any SAR they had on the target. And they said that success came from looking at this structure permeability relationship. So they were measuring the passage of arabinose into the molecule as a surrogate to see how well their compounds could get into the bacteria. Another one here, that if people really want to find new bugs, these are places to look. Combinatorial processes didn't lead effective drugs. Should we look around proven natural scaffolds, and that could be the solution to antibiotic resistance. And what can we do to re-engineer some natural products? And protein engineering, biological engineering is quite important these days. Just some thoughts here from some of our own work and work that people at UCB with Malcolm McCoss have done, where they looked at the emergence of new ring systems in drug molecules, and actually the rate of incorporation, if you like, of novel ring systems in drugs isn't as great as you think. 2/3 of all small molecules in clinical trials comprise only ring systems that are already found in marketed drugs. And this has been found elsewhere. Some of the stuff in one of our later papers, Paul Leeson, has shown that you do see the same thing. When we were looking at compounds that are so-called pseudo-natural products, which is Herbert Waldman's concept, where you can recapitulate fragmentation of molecules and you can build them back together in ways that nature is unable. So there are different ways of exploiting natural motifs and natural molecules. These are the ways of predicting natural product likeness. And from one of these papers, from the top one, it shows you how drugs and natural products are quite different to the synthetic molecules being produced in drug discovery. So drug discovery in general, and I think antibiotics in particular, we need to take heed of this about what nature can teach us. And as we get towards the end, this is from one of our more recent papers. So we had the hypothesis in time and place for nature, and then we worked with the folks from Kemble to put a data set together to look at the difference between leads and drugs and pull triage this into ways to see how far compounds got. And the coloration here is depending whether the compound is a natural product, and we know that natural products have completely declined in drug discovery. We've looked at compounds that are natural product like, so they're kind of recognized to be modified natural products. But what made Herbert jump up and down with joy is when we saw that what has really increased in compounds in recent years is the status of compounds in the high throughput screening era that are actually pseudo-natural products. So chemists have been making these compounds without realizing it. So if we can understand the concepts of breaking down natural products and recapitulating them with different connection, we may get a chance to focus on if you like, pseudo-natural products. And the success is there. And within this, there is a tentative signal that compounds that get to the clinic versus all other compounds made against the targets are more successful. So it's kind of shown you that there is, if you like, an element of natural selection in the compounds that make it further in the clinic. So, this natural by design or pseudo-natural by design is a good paradigm to try to understand. And I just like to use this. There was a wonderful series of adverts by Heineken in the 1970s for their beer that you would get a thing like an ugly caterpillar. There were all kinds of things that were having problems. They drank a glass of Heineken and the caterpillar becomes a butterfly. So, and they would always say that Heineken refreshes the parts that other beers cannot reach. So what I suggest that if you really want to reach the parts of drug space And I think that log D against the size, I didn't explain this earlier, it's calculated molar refraction. It correlates with molecular weight, but it's not perfectly the same. It's a physical property of molecules, and it does equate to size. And these are the regions, if you like, of drug space that you may want to try and find. And if you truly want to go to these regions, I believe you need to use nature to help you to get transporters, to get you across membranes, to get you inside bugs and bacteria, and so on and so forth. These are the ways that you will have success in finding new antimicrobial compounds. So if you use natural intelligence, you can access these regions that synthetic molecules, which will typically need to be up here, cannot reach. So hopefully there's a few lessons and some learning in here. You know, you might need to be brave and go against the prevailing wind and be prepared to think differently. Just think how Chris Lipinski and Franco, what they did post HTS, and you know, they brought, you know, an end to the bad practices that were going on. Do focus on your data and look for patterns and be objective. Make sure you're focusing on efficacy and not potency. And you may just need to be decisive if your translation of your series to bug killing is rather poor. Drug discovery is incredibly difficult and compromises need to be made. And I'd say don't try and work against nature. So Jim Black, Nobel Prize winner for what he did with cimetidine and many other, you know, the transmembrane space, he would always say that the best place to find a new drug is to start with an old one. And maybe we just need to add that we might want to start with a natural product or a deliberate mimic of a natural product, displaying precedented motifs recognized by nature. So think how nature can help you in your plans. So finally, just a few thank yous to many people I've worked with over the years. There's the various people in what I call the Time and Place for Nature Consortium, and how we expanded with the help of Kemble. You know, Paul Easton's been a great friend and mentor for many years, and we continue to meet in the pub maybe once a month and discuss things and put the world to rights. Yeah, there's some great insight from his work, and also my many former colleagues and friends at GSK, Alan, Mike, Chris, Tim, and yeah, far many more than that over the years. You know, you saw some work with Liz. And then finally, thanks to all of you for your attention. I'm talking here, it feels like silence, so I hope you can all still hear me. There's still plenty of people on the line, and I should thank Nicola, Susan, and the H3D team for this kind invitation. So thank you. So I've got through in hopefully a reasonable time, and we've got some time for some questions. So that's me there.
Rob, thank you so much. Everybody, I want to give as much time to your questions as possible. So if you do have questions, please raise your hand. Great to see the applause is coming through.
Yep, thank you. Much appreciated.
People have stayed on live. Salvik, you go ahead. You can unmute yourself and go right ahead.
Yeah, firstly, thank you very much for your talk, and it was very useful. As a medicinal chemist, I learned a lot. And yeah, so my question is, how do you see the FSP3 parameter in modern drug discovery?
Okay, FSP3 is somewhat controversial. And coming from the GSK camp, where we always focused on the number of aromatic rings,
right?
And that has all, and you know, and one thing that does matter is the number of aromatic rings. And if you look at, there's a recent paper by Paul Leeson he published with Kemble. I can send you a link if you like. I don't think I've referred to it in this slide deck, but he went through a big set of compounds that Kemble produced looking at various parameters, and he found absolutely no signal in that for FSP3. It's probably the biggest and best soundbite of the last 20 years, but it actually is not very meaningful. You know, there was a paper recently that, you know, claimed to be the first to point this out, but you know, GSK, we've been pointing this out for 15 years. We knew that FSP3 was rather intangible and rather meaningless. But FSP3 and aromatic rings kind of do go hand in hand. So, you know, my message would be look at aromatic rings. And then if you look at the stuff where we bring it together in PFI, this concept that an aromatic ring is bad even beyond what it does in terms of adding lipophilicity, you know, I think they interact in many ways. So if you put a heterocycle in, of course, you reduce lipophilicity. So then you can get a beneficial effect with that ring. And we see that these things are increasing in drugs. So hopefully that I've explained it in a nutshell. Use the concept of escape from flatland, but that metric, FSP3, doesn't have a huge amount of meaning, and it isn't a thing to focus on.
Yeah. Thank you, because I am aware about this controversy and stuff, and I would like to know your view as you are from a industry background.
So another thing, if I show my little mug here from Spirochem, I don't know if you can see that. So I have worked with these guys, and I've known the folks there for many years, since they started, late noughties, and I said to them, I was always worried that if you went to these kind of molecules where you've got, if you like, constraint through aliphatic compounds, that it could cause issues with reducing entropy. That's one of the reasons why aromatic works. And I said, you may just get into the same problem as you do with aromatic rings, but actually in practice, if you do use aliphatic constraint, it is good news. So it reduces it gives you specific structures, but it doesn't bring the problems that an aromatic ring does. And my explanation for that is that it's down to the many more different kind of interactions that an aromatic ring might make compared with an aliphatic system, if that makes sense.
Right. Yeah. Thank you very much.
Salvik, over to you, Matthew.
Thank you so much. This has been a really interesting talk, and I think there's a lot I'm unpacking on my mind. I think my question has changed 2 or 3 times as you've been answering Savik's question. But I think what I wanted to just get your perspective a bit more on is, it was interesting to see that model relationship that you have between ChromLogD and CMR, and the observation you made that might actually be more predictive than the artificial membrane or cellular models. And I just wanted to get a bit more of your thought on essentially how you see. So is it that there are other properties than what we can measure in this permeability assays that actually drive the actual permeability that we see in cells? Or why do you think those models are so much more predictive than, say, CACO2 or Pampa assays?
So the model goes back many years, and I look at the paper with Chris Timworth, the 2020 paper, and there is more detail in there. But it was initially a 4-box model, and they were trying to make compounds that were deliberately paracellular absorbed. So they had to be small and hydrophilic to get on board by paracellular absorption. But as time went by in GSK, they looked at the data, and they would rebuild models every year. And up until the point I left, and as far as I know, it's still the same, they look to rebuild that model every year. And Chris Luscombe used to do this, and he never found anything that was better than log-D CMR. The only step change that made the model more predictive was when we moved from oxygenol water measurements to chromatographic measurements. So if you look at the chapter I referred to, and Drug Discovery Today in 2011, which is it's in the slides, we show why chromatographic determination of lipophilicity is so much better. Basically, if your compound is insoluble, oxygenol water is a random number generator, but chromatographic measurements, they hold irrespective of the solubility of your compound. And as time went by, they would relook at the model. I was often asking Chris, what about shape? What about other parameters? And he said every time they looked, he never found anything that was better than log-D versus CMR. So it was looked at every year, and nothing ever beat it. And just, and for just pragmatic use of the compound, and experimental use of it, it was always found to outperform others. And it's a very, you can do calculations yourself, you can do predictions, and it will stand you instead. And it's actually quite cheap to get chromatographic measurements. And then CMR is the daylight version you need to use. That is available in ChemDraw. There are slightly different versions in some of the online packages, but again, it's a principle. It's generate some data, make some hypotheses, look how your compounds behave within the framework is the best advice I would give. And and in doing that, we had to show data to convince people that it was a sensible decision to stop putting every compound through the Pamper assay. I really don't like the Pamper assay. It's got problems. You know, trying to get compound back, you get things that go through little pores. You get it's not a perfect bilayer.
Thank you, Matthew, for that question. Last question from Franco, of course, and we'll just squeeze it in for 1 o'clock. Over to you, Franco.
Yeah.
Hi, everyone. Rob, thank you very much for always a great job.
Thank you. Grazie,
Franco. I'm not in Rome, so that would have been easy. I'm in Boston, and I woke up at 6 jet lag, but I didn't want to miss your talk.
You're too kind.
No. It's very well deserved. 2 things. One, thank you for imparting upon us the fact that we have to have compromises in optimization, and it's a matter of homeostasis of a comprehensive package, not one thing or the other. Mm-hmm. The other concept, efficacy, not potency. Yes, we need to keep going with that and spread it all over the world. And the last one, I would appreciate if you could send or make available the reference. You spoke 2 questions ago about the SP3. I agree that it's probably as a descriptor not a very good thing. I mean, the idea of the aromatic or not, as you have in your SFI, etc. Yes, but the FSP3 as a descriptor may not have a place. If you can make available the reference you mentioned to the, I guess was the first question, that would be great.
If I just give me 1 second. If I just need to. I assume you can see on my screen that I'm looking for things.
Yes,
it's in here. I've got a different. I'll move it across so I don't give everything away. I do have it where I'm looking here, and I will put an extra.
Thank you.
I will put this slide in at the end of the deck if people can see that. If I'll put this in as an extra slide, right? Actually, I can do it now.
Thank you very much.
So yeah, that will go in. So that's done on the fly. You'll see there are a few extras in here.
Yes,
I was half anticipating question. So Nicola, I will send you a new PDF when we're all done, and that is in there. So this is Paul Leeson's paper, and then you will see. I probably do want to put. I need to put the Fort one because there were some other things that Tim Ritchie was always very upset that there are far more references to to Escape from Flatland with its soundbite than there were to his work on aromatic rings.
Thank you.
Perfect. We've just gone over the hour, so I'm going to end off here. And Rob has very kindly offered to share his slides, as you've heard, in a PDF, so I'll be sending that out to everyone who attended. Robert, just remains for me to say a huge thank you for your time and expertise. I know this has been really exciting for a lot of our attendees to hear your experience and to get that kind of massive overview. And,
I hope it's helpful.
So much.
It may not be perfect, but I think it's a challenge. And think of Einstein. Let's not go back to how the methods that got us there. Perhaps always think.
100%. And then to all our participants, thank you so much for joining. I put some links in the chat there to our free drug discovery course if you're coming at it from a different discipline, and also to give some feedback via our feedback form. Yeah. Thank you so much, everybody. Yeah. And yes,
I should put a plug in as well for the EFMC. If you're doing drug discovery training, I've been part of a group there where we're producing similar material. So I should have done that. I should be told off for not putting it in my deck.
Well, this is the opportunity. Send me all those links and I'll email our attendees. So that's no problem.
Perfect.
Thank you so much, everybody, and see you at the next one.
Yeah, thanks all.
Thanks a lot. Bye.
Bye. Bye.